mask off for this. So welcome everyone to the third annual NORSE Roundtable at the American Epilepsy Society meeting. People who are here, please go ahead and grab all your food and drink. And those of you on Zoom, grab whatever food and drink you like as well. So this is our agenda for the day. I'm going to start with me, followed by Nora Wong, and then we'll have a data blitz. And then the second half is largely going to be a discussion brainstorming session. So we're about 2 minutes behind. So let's get started. So I'm going to give you a very rapid update on the prospective observational study that's been going on for a couple of years now. So some changes in the last year or so in what we've done. So we lowered the age limit from 6 to 2. We got Spanish consent forms. We expanded our specimen collection protocol to include not just one blood draw initially, but multiple ones, including later ones after discharge. We've added options for collecting PBMC, saliva, nasopharyngeal swabs, urine, stool. We've simplified the brain specimen protocols. We've dramatically simplified data entry by popular demand, where almost everything is entered in one data sheet now at the end of their stay rather than a daily entry. Our target enrollment is still 100 patients. I'll tell you how many we have in a minute, but we are going to keep enrolling until we have at least 25 pediatric patients. So far, pediatric enrollment is way behind the adult. And we're going to fold some of the analyses of samples into a new biorepository that I will tell you about momentarily. So as of now, we have 25 sites in our study. We have 49 patients enrolled. These are the sites and how many they've enrolled. I don't know if you can see it too well.
Larry, we can't see slides. Are you sharing or?
Oh my. Thank you.
Yeah.
Thank you for mentioning that. You guys working on that? In the name of time, I'm going to keep going, but yeah, this is a slide showing all the centers and enrollment. So, oh, there you go. Let's get it in slideshow. I'm now looking at my laptop also. So, all right, so there's the enrollment. I don't think you missed anything important on the slides before that. I said everything. Just some basic results of what's in there now. So the age range is from 6 to 83. We only have 5 pediatric patients. 68% of patients are female. Average hospital stay is a month. 36% of patients qualify as FIRES, so they had the prior fever, including 4 of the 5 pediatric ones. And we have an 18 to 20% mortality. As far as some basic lab results, so CSF, we have from results from 33 patients, about half of them have more than 10 white cells. Some relevant antibodies, so their NMDA receptor in 5 patients. There was a high level GAD titer. Actually, there were low level GAD titers in 5, but 1 had high level and was thought to have anti-GAD encephalitis. Interestingly, there were no voltage-gated potassium channel complex, so LGI1 or Casper or any of those, which is different than our old retrospective series. I'm hoping and imagining it's because we're identifying it earlier and treating it better, so they're not developing refractory status, but I don't know if that's the case. In the 4 PEDS cases that we have the data on, there were no antibodies or etiologies found. There's some positive mycoplasma titers and no infections found. Immune therapy is given to the majority of patients, but not all. And you can see a list of what they were treated with. As far as etiologies, about 80% have no etiology. NMDA is the way in the lead for identified etiology. And you see there was one CNS lymphoma and one anti-GAD. So the biorepository that came from this, currently these are the specimens we have received and are storing. So you can see we have blood and CSF, mostly CSF from 20 patients. And we just recently started collecting the other things. We have 2 whole brains and 2 biopsies as well. So that's what's in there now. So now I want to tell you about the new NORSE FIRES open biorepository at Yale. So this is a funded project thanks to the Wongs. It's basically funded for 100 patients in 5 years, roughly. It's kind of the estimate. And the idea is, I'll just move that on, is to collect, not only collect, but also do some basic analysis, store the results, and share both specimens and results widely to any appropriate people. So the idea is to be able to take samples from anywhere in the world with somebody with NORSE or FIRES without any pre-existing IRB or contract. So it's all just consented on the fly. And then we want to have standard protocols for collecting, analyzing, storing them. We are aware that other centers want to do this, and that's great. We just want to make sure that we have the same protocol so we can combine data in the end. So I think very likely there'll be a European place and probably at least 2 in the US. And then again, all the key point is that all results will be made available. So this will be an open research database. And then we will continue to have some funded projects where people will use the samples from this biorepository, but one of the agreements will always be that they have to share the results back to the repository in the database. We will be taking care of all the practical issues and consent on the Yale end and payments for shipping everything, entering in a database. We are going to fold in any remaining specimens from the current study into this new biorepository, but we will continue to get really detailed clinical information on those 100 patients in the current study. There is a steering committee that's going to help decide all those things, like who gets the specimens and how exactly to store. And we've had 1 meeting so far, but you can see the members of the steering committee, most of whom are either on Zoom or in the room. And this is the testing, the standard testing we're going to be doing on all the patients or all the samples we get into this new biorepository. So for blood, we're going to be doing autoantibody testing. Actually, we've done the CSF as well. That'll be done through Michael Wilson at UCSF. We're going to be doing probably whole genome sequencing through the Yale Center for Genomic Analysis. And we've budgeted for storing that in the proper way to share it with other researchers. We're going to be doing cytokine analysis, probably at Yale, maybe through a company, but that'll be blood and CSF as well. And then some things that are not yet funded, but we might do, especially if people are interested and have other funded projects, is more on the omics and injury markers. We do have funding for some single cell RNA sequencing, again, both in blood, CSF, and in brain tissue. We have budgeted for some brain biopsies and for some autopsies in whole brains. You'd see 3 to 5 a year of those. And then as far as we are going to collect other specimens, so nasopharyngeal, saliva, and all those other ones, but for now we're just storing them. That's not budgeted to run any analysis on that. We're already, there's at least 1 funded project already going to use the stool specimens for a microbiome study with separate funding. We have not funded iPSCs. That's something we could potentially talk about later in the discussion. And as far as clinical data, there's all that's really mandatory is a single 1-page sheet of clinical information to start, but we will also be getting copies of medical records and doing some basic follow-up of these patients later. The exact details not completely worked out yet. We do plan to get MR images for sure, and probably EEG tracing, so we can do all kinds of analyses, including big data that you might have just heard from in the presidential symposium. We haven't included control in this current budget, but there are a lot of different sources of controls depending on exactly whether you're talking about genetics or RNA-seq or something else. So it will depend on the project. I think I covered all that, and particularly want to thank the Wongs for funding all of this. And I think with that, do I have time for questions? Any questions about the biorepository before I go on to a couple other website updates? Yeah, wait for the microphone so they can hear you. Tell us who you are, Eric.
So, Eric Rosenthal, Mass General. So if we had a patient we want to refer to the biorepository, it takes sometimes time to start collecting. What's the earliest thing that one would need to do to not miss a data point?
Usually it means freezing the samples as soon as you get them, the earliest samples that you have. But we can do it. We're going to be able to do the consent for this very quickly. So I don't think there's going to be much delay in that regard. But yeah, if you're doing spinal tap and you know you might enroll them, just freeze some extra for. Jorge, is that accurate? For everyone in the room, that's Jorge Cespedes, our coordinator. Probably all of you have emailed with him. Get to meet him in person now. All right, are there any questions in the chat? Let's see.
I get Larry Levy, just one point about single cell analysis in spinal fluid. And for sites that are equipped to do 10x, like the MGH and UCSF, where these are routine things being done. If there is, as we've done for other protocols across different sites, we could potentially, if there is a patient going to have a spinal tap, if someone just emails me, we could potentially discuss how to do that, because it can be, they can be done at the local site in terms of the first step, and then the libraries and sequencing to avoid batch effects can be done together here at the site at Yale.
Great, thanks, David. You guys can email me and I'll forward it to, I'll get it to David. That was David Heffler, by the way, if you don't have his contact information. So you can always go through me to reach anybody. All right, so in the name of time, I'm going to move on to a couple other updates on the websites. I want to remind everyone of the Family Registry. Tennille will be talking about that a little more, but please encourage all of your families, either the patient or their families, to enroll in this, and anybody can. I'll let her tell you more. Just to remind everybody, some of the past awardees and ongoing studies that have been funded by the Norse Institute in some way. So this is Anna Maria. You'll be hearing from her group about this study shortly. These are some of the prior ones: an epidemiologic one, an animal model, and the last one from Olga you'll be hearing about today as well. And then going back in time, this one on genetics that's still going on in the microbiome study. And this one from Mayo Clinic is using a lot of the samples from our current study. So some website updates. One, there's links to the recordings of the scientific symposium we had last March and to the family conference we had around the same time. There's a brand new downloadable brochure. Those of you here in person, they're over, hopefully many of you are, yeah, I see many of you holding them. So it's a very simple 2-page printable brochure that's great to give to the families and patients in the hospital. And here's what it looks like. I'm not going to give you time to read the whole thing. Other things on our website, for those of you who haven't been there recently, so we have the diagnostic evaluation. Most of these things have been updated pretty recently. There's a very detailed status epilepticus treatment algorithm. We have a brand new, as of yesterday, posted annotated references, now has about 130 references on there. Special thanks to Zubaida Sheikh and Jorge for doing that, and Andreas van Balen for starting us off by sending a bunch of the pediatric papers for that. I'm going to show you some screenshots from that. There's lists of grant opportunities, our prior research bulletin, ongoing studies. There's a great family discharge planning guide on there. Thank you to Erica for that. And there's lots of pages with lay descriptions of what NORSE and FIRES are and all the terms used. There's some links to podcasts. There's information on palliative care and the Family Registry, of course. This just gives you a screenshot of the new annotated reference list. So you can see it's categorized into, they're listed in this order. So the first ones are consensus statements, guidelines, and definitions. Then there's a bunch of original research articles. There's about 60 of those. Then there's systematic reviews and meta-analyses and other reviews, case series, etc. And then within each category, they're listed chronologically, most recent first. And we've bolded some of the major articles. And then Zubaida and Jorge did this great labeling upon the polite request of mine. There were so many different ways to categorize these. And so we basically, all the pediatric studies have this little pediatric icon, and the ones with ketogenic diet have a little avocado icon. So you can easily look through and get everything you want. It was too hard to categorize things in all different ways. So you'll see like this. Here's a couple of the consensus statements. See, the first one is all pediatric. You only get a pediatric symbol if it was all pediatric. Here's some of the original research articles. You can see little weightlifting guy is the marker of long-term outcomes. And this is some of the systematic reviews. You see the ketogenic diet symbol there. And some of the recent case series. There's a lot of great stuff. If you have not checked in literature really regularly, take a look at this, because there's a lot of interesting recent articles. So before I forget to thank people for helping put this together today. So Nora Wong, who's going to speak to you in a moment, but she's really been the driving force behind all this for many years now. And it's given us lots of motivation and tons of time and effort as well, and pretty much manages the website. So Jorge, I mentioned, Zubaida, Ronny Wickstrom for leading us with the Delphi, and you'll hear all about that in the second half. Tennille for the Family Registry. Nicholas has helped with a lot of this, including the annotated references. Thanks to AES for letting us have this in conjunction with meeting every year. The rest of our medical advisory board. All the patients and families, obviously, if they don't consent to this, we can't do any of it. And thanks to the other speakers, including those from other countries, some of who actually made it here, and some who will be on Zoom. So with that, I'm going to turn it over to Nora.
Thanks. Welcome, everyone. I'd like to address two questions you might have right now. What's the intention of this biorepository, and will they really share samples and results? When Anna Maria Visani joined our medical advisory board in early 2019, I learned from her that the aim of a biorepository is not simply to collect samples, but to have them analyzed and interpreted. And because no one knows what NORSE is or the best analytical tool for its study, different analyses should be considered. No single approach should dominate. For samples to be usable for a variety of analyses, a sampling protocol had to be developed with that end. Anna Maria enlisted the help of her colleague, Eleonora Aronica, neuropathologist from Amsterdam, to introduce that challenge to us at the NORSE Roundtable in 2019. Eleonora joined our board shortly thereafter, and our board began to prioritize research questions and the analyses needed to answer them as a means to develop an actionable framework for research. Anna Maria, Eleonora, and Larry are leading the development of a standardized sampling protocol beginning within our board. Larry has been balancing these various very strong viewpoints with the realities of Yale and our budget with great skill and diplomacy. The result is the plan he just presented. The biorepository is conceived to be a living project, changing in response to its findings and that of the larger community. We are not trying to compete with other repositories. As you can see from our recently expanded Medical and Scientific Advisory Board, we have asked people we know are collecting samples to join our board to increase the likelihood that our collections are harmonized from the beginning and comparisons can be made across repositories. In regards to our sharing the results and samples, I point to two areas of precedence. First, at Yale, David Hafler, Chair of Neurology, is working on this project. He made his landmark single-cell RNA sequencing analyses in MS available to others on a public data platform. We will follow his precedents. Chen Liu, Chair of Pathology, who is also part of this project, arrived at Yale in 2020 with a dream of establishing an open, collaborative libraries of disease with people contributing samples, other people analyzing things, and then the results to be shared again. There is solid commitment to make the NORSE FIRES biorepository one of the first open libraries of disease at Yale. Second, look at the history of the NORSE Institute. The first international conference on NORSE and FIRES in Salzburg in 2017, and the AES Roundtable meetings since then have been organized with the aim to gather investigators in the same physical space to exchange ideas. In 2018, our board, all clinicians at the time, made the decision to issue an RFP through NORD for $100,000 with preference for translational research because we knew that we needed scientists to join us. Chuck Howe and Eric Payne were awarded that grant. Nicholas and Larry have arranged to give them biosamples from the consortium study for that project. Chuck and Eric are now part of our board. Our aim for the biorepository is to provide preliminary results you can use to apply for NIH funding or other large grants. Vicki Whittemore from NINDS is here again this year. She was here last time to answer questions you may have about collaborative NIH grants. And Vicki, can you raise your hand so everyone, there she is. To get the results you can use from this biorepository, you need to not only send biosamples, but you need to inform us now of analyses, sampling, and storage issues vital to your research. This is the goal of the biorepository. So we hope you join us. Thank you.
Thank you, Nora. We're now going to jump right into our data blitz section. Our first presenter is Malika Sarovic from Anna Maria Vizzani's lab in Italy, who should be on Zoom. If we could switch to her.
Can you hear me?
Yes.
Okay.
Larry, if I can make just one quick point about data sharing, because as you all know, once papers are published, data is required to be shared. But what we want to do is to share the single cell data in real time with the group. So as we generate those data, the raw data files will be available to anyone in the group, particularly in relationship to their samples or others. So which is somewhat different from other types of sharing that we traditionally do. So just to be clear about that. Sorry for
Great.
interrupting.
Yeah, thanks for clarifying. It's great.
Okay. So we're just putting the slides on. So, hello, my name is Milica Cerovic, and I will give you a brief update on our development of in vitro model of FIRES for rapid selection of effective drugs. So, our in vitro model is an adaptation of an already existing, of an already validated model of drug-resistant epilepsy, where the epileptiform activity is elicited by zero magnesium and 4-aminopyridine. So in our experimental setting, we are using horizontal brain slices from mice, which are 28 to 30 days old, which roughly corresponds to school age or early adolescence in humans, according to literature. So these slices comprise renal cortices, hippocampus, and subiculum, and we are recording them on microelectrode arrays, where the activity from entire slides can be monitored simultaneously. We see the activity as in form of synchronized local field potentials that can be single events or repetitive events. So I will show you just two important patterns that we see in our recordings. So if, for example, LFPs can occur with in high frequency bursts, and if they are more than 3 seconds long, they are classified as ictal events. So to interpret this kind of raster plot, consider that on x-axis there is time, and every LFP detected is a dot in the graph. So the vertical lines, what resembles vertical lines, are the synchronized LFP events. Another important pattern for our research is this, what we classified as status epilepticus-like events, where the LFPs are occurring at a sustained frequency, at least 1 Hertz, for long periods, such as 5 minutes or more. So to make the this model more relevant to FIRES pathology, we added an inflammatory component. So we pre-incubated our slices with lipopolysaccharide, which is a prototypical neuroinflammatory agent, and for 30 minutes. Then the slices were briefly washed out and recorded for 60 minutes in presence of zero magnesium and 4-aminopyridine. So we first wondered whether this kind of pre-incubation is sufficient to induce neuroinflammation in our slices in the time frame of our experiment. So we performed real-time PCR analysis of inflammatory markers and cytokines, and we saw actually a prominent inflammation. I will show you only 2 examples of the markers that we have measured. We can see that tumor necrosis factor is increased in 1 hour after LPS removal. And importantly, we can see that the ratio between inflammatory cytokine IL-1 beta and its antagonist of its receptor antagonist, endogenous, this balance is shifted, strongly shifted towards inflammation. So this confirms to us that the model is adequate for testing neuromodulatory drugs such as Anakindra. So in terms of epileptiform activity that we saw the with the LPS, we saw an increase in the activity, and it could be seen as an overall increase in LFP frequency, but most importantly, we saw an increase in the incidence of status epilepticus events. So they were quite rare in the classical model, but in the one when we added LPS, it became frequent. So we tested we next tested whether the anti-seizure medications at therapeutic concentrations, such as phenytoin, sodium valproate, and phenobarbital, can modify this heightened activity and or whether they can inhibit it. So we used the same protocol and allowed for 10 minutes of the epileptiform activity before adding our anti-seizure medications. So what we saw is still work in progress. So we have to do the quantifications and increase the number of slices. But I will show you here what typically is happening in a slice, and with an example of phenytoin. So in the panels A1 and A2, you can see that the activity before adding phenytoin, 10 minutes of activity, there is some epileptiform activity that is reduced after 40 minutes of phenytoin. It is strongly reduced, but it's still present. And the far left panel, you can see the same thing, but with a Faust color map graph, where the brighter the color, the more frequent the LPSs are. So when we add the LFPs, sorry. So when we add the lipopolysaccharide to our model, there is some variability, but in the cases like this one that I'm showing you, where the sustained activity is already induced in before adding the phenytoin, we can see that this kind of activity cannot be inhibited by phenytoin application. And we still see after 40 minutes of the application that status epilepticus in hippocampus and sustained activity in cortex. And the same pattern we see with valproate and phenobarbital. So in conclusion, we saw that epileptiform events are exacerbated by lipopolysaccharide-induced neuroinflammation. That this neuroinflammation increases incidence of status epilepticus-like episodes, and they're refractory to anti-seizure medications. So as a next step, we will test whether immunomodulatory treatments, in particular Anakindra and tocilizumab, can inhibit this kind of drug-resistant activity. And if they're not effective on their own, we will combine them with the already used anti-seizure medications. In the end, I would like to thank the NORSE Institute for awarding us with the seed grant, which allowed these experiments. Thank you. That was.
Thank you. That was fantastic. We do have microphones we can pass around. People who have questions, just raise your hand and someone will bring a microphone to you. But that's fantastic. It's actually see a model specifically designed to mirror NORSE and FIRES, and then testing treatments. That's exactly the kind of thing we're hoping for. Nicholas?
Thanks, excellent work. Are you planning to get pathological data on those animals, especially to look at in the colostrum to see if there is specific damage there? Because it's actually the only thing we seeing might be specific from in NORSE cryptogenic NORSE cases is its colostral hyperintensity on MRI. So I would look at pathological data and see if there is increased cell deaths, for instance, neuronal deaths in the colostrum.
I'm not sure that I'm getting that pathological data from the animals.
Yes.
Right. Okay. So well, this is in vitro model. So definitely what we're going to do is to pass to in vivo model when we have our best candidate. And then we'll see how the pathology in vivo is evolving, and whether we can block it with the drugs that we have chosen. If that is answering your questions, I'm not sure.
Dr. Hafler had a question.
Yeah, just to follow up, it's very interesting. It's a very nice model for drug testing. It'd be really interesting and to ask the question whether this involves activation microglia. There are different states of activation of microglia where they can be more suppressive or active. And the question would be whether there's the synaptic pruning associated with LPS activation of microglia, which reset the cycling. And they require very specific expertise, perhaps optogenetics and other techniques that have been developed. But I think just in terms of whoever has the expertise in mice to look at that, the whole idea of microglia activation, of synaptic pruning has become just central in the field of neuroimmunology. I think it'd be really interesting to look at those factors with a model like this, associated with the resistance to drug therapy for the epileptiform activity.
I'm just not sure whether in this such a short time frame we could look at the synaptic pruning, but probably if we give the neuroinflammatory insult in vivo and then look at that in the slices, then that might be possible and interesting, definitely.
Yeah, I have no idea what the kinetics would be for it, but it's a beautiful model for potentially. Now, I mean, the purpose of the model should inform us as to what we should look at in humans, because you can manipulate the model in ways we can't do in humans. So hopefully you can give us clues as to where we should focus our attention pathologically and from single cell analysis.
Great. One quick question. Do you plan to look at steroid effect also? I saw you had specific treatments, but how about our most common treatment of just steroids?
Yes.
We're not planning at the moment, but I'm not sure. That maybe Anna Maria could answer this one.
Yes, we are thinking actually to try methylprednisolone that, if I well understood, does not really work very nicely in the clinical condition, to see whether the activity is resistant, this in vitro activity is also resistant. But of course, we can try also other steroids. So we would be happy to have suggestions about drugs to be used to validate the model as better as possible.
Great, thank you. Any further questions, please put in the chat and you guys can continue the conversation there. But we're going to need to move on in the name of time. Next, we have Tennille Gofton. We'll be talking about the Family Registry, also on Zoom. If we could switch to her.
Thank you very much. And I have to say, I'm very impressed with how this is working as a blended in-person and over Zoom. So thanks very much. My name's Tennille. I'm working at Western University and with many people who are present or on Zoom also on the NORSE Family Registry. And this is a project that's been very generously supported by the Cohn family. And so we thank them. So the goal of this family registry is to provide a systematic place for families and survivors of NORSE who are not necessarily connected to a research institution, to be able to contribute to a registry to help us learn about people affected by NORSE worldwide. And the goal is to inform future research and also learn more about outcomes. So essentially, some of the questions that I think we'll be able to add to the literature with this is whether or not there's a particular worldwide distribution of NORSE or FIRES, or at least where we can get participation from. And then we're hoping to look at also whether the short-term and long-term consequences can at least be outlined and provide the hypotheses for future research. So just quickly, how does this registry work? For those of you who haven't heard me talk about it before, it's based in REDCap. And so people who are either survivors of NORSE, if they're functioning well enough, they're surrogates, or their healthcare teams can enter data directly into REDCap, and their consent is actually done electronically through the REDCap system itself. So they don't have to be connected with an academic institution, but they do have to be at least web savvy enough and able to connect to the internet. If families or surrogates don't want to, then they can also have their healthcare provider input data, and there's a consent process for that also. So the information collected is some demographic data, some information on presentation and hospitalization, but also some background risk factors. We're looking also at some diagnostic evaluations and hospitalization details. These are mostly to help confirm that the participant did have NORSE or FIRES. And then we're also looking at long-term outcomes, and doctor-family communication and survivorship, as well as quality of life. So some of the important features, I did mention that written informed consent is required at the beginning, and this is used or actually obtained through REDCap. REDCap then, after the initial survey is completed, will send automatic links to an email that the person doing data entry identified, and this is to help us do follow up with a very short survey at 6 months, 12 months, and 24 months. There's a possibility for people to save and return if they're not able to do all of the data entry at one time, because that certainly is an issue for people who are surrogates entering data, and also parents or caregivers for somebody who has survived but requires more support from NORSE. So we have translated it into 4 languages at this point in time, and we're hoping to increase the number of languages to increase our outreach. In fall of 2020, there was also a revision to incorporate more detailed outcome measures, and so that's been ongoing for just over a year. Briefly, we have about 69 participants from 12 countries if we look at all languages. The mean age of participants is 26, ranging anywhere from 3 to 78, and 55% are male. The most common symptoms at onset are flu-like fever and headache symptoms that occur in the first 7 days. And in less than 25%, seizures were the first presenting symptom. And our mean ICU stay in these individuals was 44 days. If we look at the number of people who are survivors, we have, this is a slightly older slide. I wasn't able to abstract the data recently this week, but 25 out of 45 in late September were survivors of NORSE and FIRES, and about 19 of those people had presented in a manner that was consistent with FIRES with a preceding febrile illness. 27 out of those 45 had been informed by the medical team regarding NORSE and FIRES, and 28 out of 45 had actually heard about what their prognosis was. And about 37% of patients had had the palliative care team involved. And I don't have this data here, but if you look at satisfaction with communication between the healthcare team and the substitute decision makers or surrogates, it was really all over the board. Some people were very satisfied and some people were not at all satisfied. If we look at outcomes, this is where we start to have a lot less data. I'll come to that issue in a few minutes. Survivors that have reported their outcomes have quite impaired outcomes from the point of view of seizures. There's usually greater than 10 seizures per month. All survivors that we have on record have had epilepsy, and all of them are on multiple anti-seizure medications, which isn't surprising. This is actually the data point I find that is the biggest moving target when we look at the data over time. So we've been looking at quality of life using a visual analog scale, with 0 being the worst outcome and 10 being the best quality of life. And this number has been changing quite a bit over time, but at last check, it was about 3.9 plus or minus 2. And at some other times when you look at the data, the scale kind of slides up and slides down. So this seems to be a point that changes quite a bit with data entry. So challenges that we've run up against, I think these are important. And Carnegie, the research associate who has been doing the lion's share of the technical work for this, and I are going to sit and look at these in January 2022 to see how we can troubleshoot them, but any input or feedback is welcome. Our initial survey completion is high, so looking at the initial hospitalization and outcome first hospitalization. But our follow-up survey completion for any of the recent survivors are quite suboptimal. And some of this is because enough time hasn't elapsed yet for them to be eligible, but some of this is because I think they don't have time to go back and complete surveys, where it's very difficult for them to on an emotional point of view. And so that has led to suboptimal data right now from the point of view of actual outcomes. Interestingly, our follow-up surveys are very, very short compared to the initial survey, but I think people don't know that when they're going to be going to do it. The second thing is there's currently no requirement for a physician to complete some of the information about the hospitalization. When we designed it, the data fields are quite basic from the point of view of the information they're asking, but that is also from the perspective of physicians. So for families, some of these questions might still be pretty overwhelming. And so we're planning to look at how we can address this in January 2022. Written into the consent form is the possibility to twin and pair with data from other studies. So if there are people that have participated in the NORSE Family Registry and they have participated in the NORSE, the repository, then potentially we can combine some of that clinical data to help strengthen what data we have. And frankly, I wonder if the survey might be a bit too long for survivors when their caregivers can't find the time to fully complete the initial survey. So our next steps are really to troubleshoot on the issues that I just outlined, but also to promote registry participation, increase the number of languages, and try and increase our reach. I think we were, as with everybody in the world, delayed a little bit with our attention diverted during COVID, but I'm hoping that we'll really get back to this. And we also have other thoughts about how we can link and bring physicians into inputting data by potentially giving them a separate link in REDCap they can use. So anybody here, if you can promote the registry to your patients and their families, the link is available through the NORSE Institute website. And we're totally open to any other suggestions that you have. So thanks very much to everyone who's contributed to this and for your attention.
Thank you, Tennille. Take questions or comments.
I don't see any in the chat. Oh, I'm watching. Do we plan to translate into German? Yes, we do. That is something that we have been working on is our next list. So our current languages are English, Spanish, French, and we have a Chinese character version that should be suitable for both Mandarin and Cantonese that is about to go live. And after that, we're trying to hit the languages that are most frequently spoken and will target the largest populations. Any relationship between outcome and feedback regarding information provided by clinicians? That's a good question. We haven't done that analysis yet. From the point of view of whether we'll be able to detect an association, I don't think our numbers are big enough quite yet of people who've had good, rated their discussions about prognosis with the healthcare team as well or good. And also learning about their outcomes. So I'm not sure we'll be able to pull that data out yet, but we do still have a few more years to collect data. So participation is not limited to families of survivors. The family of deceased patients can also participate. So the registry is built so that the questions do change based on what your responses are. So if somebody has passed away, then they don't see the portion of the registry that is meant for survivors. So please encourage them to participate also if they're up to it.
Fantastic. Thank you, Tennille.
Thank you.
You can answer any further questions in the chat. So you've heard from Italy and Canada. We're now going to move to Belgium and hear from Claudine Sculier. Claudine?
Yes, hi everyone. I'm going to share my presentation. Yes, you can see it. Okay. Okay. Hi, first, thank you so much, Larry and Nora, for inviting me to this session. And I'm really sorry not to be on site today to present this study. We started a few years ago with Christina Barcia Aguilar, Nicola Gaspar, and Toby Loden-Kemper with all the PISR group. I want to thank again here. So this study aimed to describe the characteristics of children with NORSE, with the clinical profiles, etiology, and outcomes. It was interesting as the previous studies on NORSE mainly involved adults, and also the novelty of this study was to use the new definitions of NORSE and FIRES. And as you know, there is no criteria of age anymore. So you will see this study is also about terminology. And just to remind, prior description of NORSE and FIRES were often based on variable inclusion criteria and terms. In particular, most FIRES studies used to include patients with criteria of prolonged or super refractory status epilepticus. And here we did not use any criteria of severity, but except those definitions. So this study also intending to kind of test those definitions. And regarding the FIRES subgroups, we also want to assess the effect of fever occurrence on the outcomes. So we created 3 subgroups for that: NORSE without fever, FIRES, and febrile status epilepticus. So the method was based on those definitions. I'm not going to read them, as you know already very well. So we worked on the prospective multicentric PCERC cohort. Those patients, those children are included if they are aged from 1 month to 21 years, and if they present focal or generalized seizures turning into refractory status. From those patients, we select those who fulfill the criteria of NORSE definition. So no history of epilepsy or the other neurological condition, and without any easily identifiable cause of status. So the PCERC database was at that time collecting 279 children. 92 of them had a first refractory status. Then from them, 49 had no easily identifiable cause of epilepsy. And finally, we got a cohort of 46 NORSE cases. We made two main groups. Those with an etiology identified, and there were only 6 patients, and a group with a known etiology, previously called cryptogenic, so 40 patients. And this group was, of course, our group of interest, and we subdivided it in 3 categories according to the previous febrile illness, which is used in the FIRES definition. So the first group, those with patients with fever present at status onset, so starting within the 24 hours from status epilepticus onset. The second patient with previous fever starting between 2 weeks to 24 hours before status onset. And the third one for patient without any fever reported. So here you can see the results. They were very different between patients with fever at status onset, so the patient with febrile status epilepticus, and patient with previous fever, so the FIRES, of course, but this was expected. So patient with febrile status were younger. Patient with FIRES had very long status epilepticus duration, and they required more therapeutic lines and additional treatments with ketogenic diet and immunotherapies. Also, when we look at investigations results, FIRES had very frequent abnormal findings, while febrile status had often normal results. And when regarding to the group without fever, they had very variable results, and we couldn't really provide any conclusion on them. Outcomes were quite concordant with previous studies on FIRES. So FIRES had high mortality, almost 20%. 25% had good outcome on discharge, but only 16% had normal cognitive outcome, and 9% were seizure-free at last follow-up. Compared to febrile status, they had 80% with normal cognitive abilities at last follow-up, and 72% seizure-free. So to summarize, this study is a description of NORSE in children with a prospective database and based on the ILA approved definition of NORSE and FIRES. The etiology of NORSE is unknown in most cases, almost 90%, so much more than in adult NORSE. The FIRES subgroup features are concordant with previous pediatric studies on FIRES. So they had very prolonged status epilepticus duration and worse outcomes, abnormal findings, MRI findings, CSF findings. And so those results were concordant with the previous studies, even if here we did not use any criteria of severity, but only the previous fever, which probably attests its very important role in this disease. And the largest subgroup is the children with febrile status epilepticus. And the question was, should they really be considered as NORSE? Because clearly this is a well-known entity with a very different clinical profile, which is less severe. But we could say this study confirms that they are separate entities and possibly brings opportunities to better distinguish them in the definition. So we know definitions in science are always evolving, and here our results probably suggest we could add another criteria to distinguish FIRES from febrile status. Like here, the status epilepticus duration, by example, status epilepticus duration more than 48 hours for FIRES, or like age more than 2 years old for FIRES. So, of course, this idea needs to be confirmed by larger studies. I don't know what you think about it. Thank you.
Thank you, Claudine. We'll open up for some questions. I can tell you in the definition, we were careful to distinguish FIRES from febrile status, but I think we messed up in still allowing it to count as NORSE. We probably should just exclude whatever qualifies as febrile status from the whole thing. I think that was the intention, but we didn't quite word it correctly.
We couldn't really avoid them in this study when we using the definition, yes.
Wait for a microphone, please.
Larry, how are you dividing the two? And also you could use the febrile status data, which is publicly available, to get some information on that from FebStat study.
Yes.
Oh yeah, there's lots of data on febrile status. Yeah, so I think we probably should publish a revision excluding febrile status, but I also do think it's a very important comparison group for all kinds of research. Suki?
When we first gathered at one of our first FIRES NORSE meeting, we decided that age should be cut off to prevent that febrile status epilepticus be excluded from NORSE. And we decided it was 2 years old, and we are right on target according to this data set. I think it was 1.7 years was the upper limit, and the FIRES was 2.4 to 8.9. So I think that was actually a good estimate from our part.
Okay.
It looked like the outcomes of NORSE with no fever were actually better than would be expected. And I was just curious if that was a reflection of the cohort and who was entered, or thoughts about that, or maybe it's different in pediatrics compared to the numbers we have in adults, but I was surprised by the outcome of NORSE with no fever.
Yes, we were also surprised by this subgroup, but it's we cannot really conclude because there were only five children, but they had variable results, but they tend to have good outcome, much better than FIRES. And so yes, it was quite surprising, but we don't know anything about them actually for now. So this could be also studied in further studies.
Great, thanks again. We're gonna have to move on. So we're now moving to our in-person speakers. So next we have Yi Chen Lai. They'll pull up your slides. And use the microphone.
Okay. Hi. First, I'd like to thank the NORSE Institute for hosting this gathering again. And it's very nice to be able to see some of the colleagues in person again. I'm one of the pediatric intensivists at Texas Children's Hospitals. And since 2017, we have sort of incorporated Anakindra as part of treatment protocols for children with FIRES. So for the next few minutes, what I would like to do is share our single center experience with you guys. With a particular focus, since I'm intensivist, particular focus on the early clinical course following the Anakindra initiations. And as I guess I would imagine that all of us as a clinicians, once we started therapies, the question has always been how to determine response, right? So is there any objective criteria that tells us that really, the patient is responding to therapies and obviously, one of the things that we're also trying to sort of figure out is whether or not there are any factors that would allow us to predict a therapeutic response even before we initiate treatment. And I would also cover a little bit about our long-term outcome data, which is roughly about a year after after their hospital actually after their seizure onset, not after the hospital discharge. So what we have so far is 10 consecutive children with that met the definition of FIRES. And what we extracted is the anti-seizure medication regimens at the 24-hour prior to Anakindra initiation and at 1 week, 2 weeks, and 3 weeks following therapies. And we were one of I think we're, for whatever reason, our rheumatologist, Dr. Eyal Muscal, actually like to send a lot of cytokine profiles and serum CRPs. And so we have a lot of the data. And so we thought maybe we can try to make sense out of the data that we already have. And we also have some of the outcome measures. And again, the long-term outcome measures is just quite crude at this point in time, which is the pediatric cerebral performance categories. And here's the patient data summaries. I'm not sure, yeah, you can see this. So our, our time interval between the onset of, between seizure onset and Anakindra initiation really vary quite a bit, up to about 40 days or so. And we're actually one of those institutions. I don't know whether or not anybody else is out there does the same, but we are the institution that will keep them on Anakindra all the way up to after their discharge from the hospital. And then the physicians will win the Anakindra as an outpatient. And, you can see here, most of the patient require, all of the patient require at least one, if not two anesthetic infusions for seizure controls. And what we actually have observed is there are sort of two different group of behaviors or progression, if you will. And so you have a group one, which is there are three patients in that categories. Really, we were unable to make any move on any of the anesthetic infusion all the way till about 3 weeks following the Anakindra initiation, whereas the 7 others, this is probably a sort of common experiences with the clinicians at the, with the clinicians that once you initiate again the Anakindra, we were able to at least win some of the anesthetic infusions. And what I did not show here is the question has always been, are we just substituting higher dose, for instance, scheduled phenobarbital for, pentobarb? And what I did not show here is the number of scheduled anesthetic medications and the level of drug levels really did not change in the group that we were able to win down on the anesthetic infusions. Now, this is basically all the data points for the 10 subjects. The one with the open circles are the one that we classify as the non-responders, the 3 that basically we couldn't make any win in the anesthetic infusions. And what's actually surprising to us is the CRP values are actually quite elevated, and these are obtained in the absence of any known bacterial viral infections. The line that you see, the black line is the reference value for normal. And so what you can see is, both the responders and non-responders actually has quite a few abnormal CRP values. And what's actually kind of interesting, and this may be mostly sort of selection bias, is the non-responders has persistently elevated CRP values up to, I guess, close to 300 days from seizure onset. And the other thing that we also sort of noticing is the non-responders tend to have higher soluble IR2 levels as compared to the responders, which then sort of raises the question of, are we actually looking at the same group of patients, right? And again, we don't have enough statistics to do any of the robust analysis for the soluble IR2 data. But what we do have is we also see that there are actually some sort of differences between the serum IL-6 and IL-10 levels between the responders and non-responders. And again, the circle ones are the non-responders. And what you can see is the serum IL-6 levels they're pretty normal as compared with their IL-10 levels are usually tend to be abnormal. And for these, we were able to do some sort of rudimentary logistic, multi-variable logistic regressions, adjusting for the presence of infection and the time to Anakindra initiations. And what we found is that the responders are more likely to have an elevated IL-6 values compared with, and are less likely to have elevated IL-10 numbers. Now, this is the last of the data that I'm going to show you, which is the short-term and long-term data, and which, if you think about it, it makes sense, right? So the responders were able to win off the ventilator a lot faster. Their ICU length of stays are shorter. Their hospital length of stays are shorter. And what we also found is that the responders tend to have a better long-term outcome classified by a PCPC. So again, these are just sort of thought process. Really, initially we're looking at basically whether or not we can find any change in seizure frequencies. But as you know, a lot of these kids are basically on birth suppressions or, has a ictal continuance pattern. So trying to sort of just do a straight out seizure frequency count may not necessarily be that helpful. And of course, what we're thinking now is, and what we suspected all along, which is what Dr. Moscow has sort of discussed in this forum, which is really, if you wanted to try Anakindra, you probably should try it for 2 weeks before deciding whether or not it's helpful or not. It's a question of really right now, I think it's too early, but whether or not there is any predictive values about the cytokine profiles. And, I'm basically showing you our sort of comparison with the response rate as published by Kramer. Those are the breakdowns. Now, we don't have any, mortalities, but then again, I'm from Texas. So, withdrawal of care is usually now sort of top in the forefront. So I'm not surprised that we didn't have any mortalities. But what I would like to point out is at least 7 of our patients has sort of normal / mild developmental delay, but really almost all of them, I think 6 out of 7 will have been categorized by the Kramer paper as a normal with learning disabilities. And so this is our future directions. Again, the question really boils down to, at least for the clinician at the bedside, is can we have a better immunophenotyping of these patients? And so whether or not we'll be able to use that to sort of as a precision medicine and help us tailor a little bit about treatment response. And certainly, one of the questions still sort of kind of, we always ask ourselves is there really any other better outcome, better markers to tell us that it's time to win, right? And so as a practice nowadays, at least in our institution, we start in Anakindra, wait for about a week or two, and then we just sort of shrug the shoulder and say, it's long enough, so let's start winning and see what happens. So is there any other sort of better EEG markers to tell us, really it is safe to win the anesthetics? So thank you very much for your time.
Great, thank you very much. Time for one quick question. It's fantastic to see a cytokine profile associated with response.
Thank you very much for this very interesting study. My question relates to the pharmacokinetic interactions of Anakindra. First of all, Anakindra itself suppressing interleukin-1, which has a downregulatory effect on cytochrome P450. This may be different from person to person. And of course, the barbiturates enzyme induction, did you control for factors like that to look at the serum concentration?
So we do have the serum. So for instance, we also have some serum phenobarbital levels. What we found is that the responders usually tend to have lower serum, phenobarb levels. And the way I interpreted it was mostly just they don't need as a high of a barbiturate levels to sort of for better seizure controls. But I haven't really sort of think of it from the standpoint of whether or not that would be a confounders, sorry.
Great, thank you again. Okay, we are about 15 minutes behind, but didn't want to cut off too much of the discussions. Fantastic. We started a little late. So our last Data Blitz speaker will be Olga Tarashenko.
Okay.
Can't point.
Oh, okay. Well, good afternoon, everyone. Thank you again for inviting me today. I have a very short talk because I've learned that Larry is very strict on time, but looks like he may have mellowed down over the years. So I'm a physician scientist from the University of Nebraska. I would like to right away acknowledge my collaborator, Dr. Xiaowei Li, who used to work at UNMC, but then recently moved to the Wash U at St. Louis, and he was a driving force behind the study. So we so this is my disclosures. I was funded by the grant from AES NORSE Institute for this work, and then I sit on the Medical and Scientific Advisory Board. We were focused on Anakindra because it's such a promising drug, and as you know, it is a recombinant IL-1 receptor antagonist, just like previous speakers have highlighted. Of course, we know a plethora of case reports and recent case series of patients who improved while they were resistant to other treatments for FIRES. And then in our animal model of autoimmune seizures, we showed that Anakindra administered to mice attenuated seizures, improved memory, and reduced the expression of inflammation in hippocampus of animals infused with anti-NMDA receptor antibodies. So it was definitely a lot of data on using this compound further. However, Anakindra has unfavorable pharmacokinetics in the sense that it has a short half-life, only 6 hours, and oftentimes requires IV route of administration, which is already scarce in the ICU. So we were hoping to develop a compound that can be administered intranasally and can directly bypass blood-brain barrier and also has a longer half-life. So that was the goal behind developing the nanoparticles. So we used this in fabrication of Anakindra loaded nanoparticles. We used water, oil, water method. And then, as you can see, we came up with successfully developed those nanoparticles. Below in the left-hand side panel, you see a scanning microscopy of the particles, and they have this nice round shape, and they were all uniform in size at about 200 nanometers. And in the right upper corner, you see in vitro release profile from the nanoparticles. And as you can see that it has been stable up to 3 days. Again, it's in solution and in vitro. However, if the same profile will be maintained in vivo, we can perhaps space out the administration to daily or even every other day. For the future studies and ongoing studies, we would like to optimize nanoparticles to extend Anakindra release in vitro to hopes that it will also transform to the same in vivo properties. We will evaluate the stability or shelf life of the Anakindra loaded nanoparticles. And then lastly, confirm the bioactivity of the release Anakindra if administered intranasally by using immunohistochemistry, ELISA. And then finally, use intranasal administration to test the nanoparticles in our autoimmune seizure model. Some things that I would like to point out that in the process of developing nanoparticles, as you can see on this last stage, in addition to nanoparticles, which comprise only 20% of the solution, we also had a lot of microparticles, which are up to 80% of the solution. And in this next slide, I put them side to side to highlight their differences. So again, on the top panel, the previous slides for nanoparticle, on the lower panel, you see corresponding data for the microparticles. So they also are of the round and nice morphology. However, their size is about 10 times larger, from not 200, but rather 1000 nanometers. And also their release profile in vitro is kind of similar, but maybe they are less stable because we can only go up to 2 days with them. The other point that was pointed out by Dr. Lee, my collaborator, that the size is quite large to administer intranasally because it will not go through the mucosal membrane, and most likely will need intracranial administration because of the size. With that, I would like to thank my mentors, Howard Fox at UNMC and Ray Dingeldine at Emory University, as well as my other colleagues, faculty who helped me with this work, as well as other projects, my supportive chair, Matt Rizzo, lab members, and my collaborators at Mayo Clinic, Creighton University, and Lankenau Medical Institute. Special thanks to Xiaowei Li, who is currently at WashU, as well as Sobi for the gift of Anakindra. And thank you again for the funding provided by the NORSE Institute. That's it. Thank you.
Thank you very much. That was excellent and precisely hit the point, because Anakindra is not the perpetrator decreasing other substances, but it's a victim and a short half-life. And the whole mixed results may be simply due to pharmacokinetic problems of Anakindra, and this can overcome it. But who holds the patent?
So Sobi owns all the rights and intellectual property because they were donors of the drug. So that was spelled out in the MTA between originally our institution and Sobi. And unfortunately, because the collaborator moved, we need to create new MTA between two institutions now. And so the whole research was on hold for a while because of this regulatory issues. Thank you very much.
All right, thank you to all our Data Blitz speakers. That was fantastic. I'm now going to turn it over to Ronny Wickstrom for the next portion of the program.
Thank you. So let's see if this goes. The idea now is to have, we're running a little late, but the idea is now to have a little time for discussion concerning, oh yeah, sorry. That's like a slide very late in the, do I skip back all the way back? Are you on the website? That's not the slide presentation, perhaps. I'd like to ask you to bring out your cell phones because the idea now is to have an interactive polling session. You can do this. You're going to be seeing a QR code that you can scan, and those of you at home can also do that. Yeah, it's running up to the start. There we go. You should be able to see the QR code. And if you scan it, you're going to get a link that'll take you to a polling site called WooClap, built in Belgium. Is it working for you? Yeah. Great.
No. We don't see the QR code online.
Oh, can we get the, how do we share it online?
They have to show. Can you share the screen?
Hang on a second and we'll hopefully have it. So what we're going to do is yeah, but that's just a general, that's not a code, no. Is it working now? No. Working on it. Still nothing? Let's give them 15 seconds to solve this. Or do we have the code so we can give them an access with the address?
It might work to just put the address in the chat.
Yeah, we need a code though. This is just a general address for the WooClap site. Let's do this. If I start, if we can go on, I think everyone here is scanned it already, and then maybe we can pop back. But you're not seeing the presentation either then?
No.
No. No. I see. There's a febrile work going on to solve the technical issues. Is it working now? Are you guys seeing it online?
Yes, yes.
Oh, fantastic. So we'll give you 10 seconds to scan the code there. 10 full seconds, yeah. All right, everyone done? Let's participate. So the idea now is to have a little discussion concerning some future research directions in NORSE and FIRES. And we're going to start off by presenting some of the consensus statements. Many of you who are in this room were part of the Delphi that we performed over the last couple of years. And we presented the data yesterday. We didn't present the research in the last section there. And I'm just going to go through that as a basis for the discussion now. And then we're going to have a discussion based on a couple of questions. Firstly, concerning clinical studies and then on lab studies, and we'll try to compress everything because we're running a bit late. So the consensus approach that we did, we did that, of course, because of lack of data, basically. But as indicated in red here, it was also an idea, an outspoken aim of this to create a foundation for future studies within a larger group, both for observational studies and the alignment of different registries, development of common data elements, and create a research consortium for future research. And most of you, or many of you, saw this yesterday, so I won't repeat it, just to say that the idea of the project was to generate a consensus guideline. And you can see at the bottom here, what the panel was. It was 48 persons, many of who are here now, mostly from North America and Europe, very few outside North America and Europe, which is a little bit of lapses from our side, of course. And what we did was basically, we'll skip all this, and just to say that following 2 rounds of Delphis that were performed in January and April this year, we ended up with the consensus statement that you can see down here. It's 85 statements, so it's very long, divided into 5 sections. And the 5th section contains 7 statements of concerning research. And that's what we're going to look at. And just to remind you again, this is what we, how it worked. So there was a 9-grade Likert scale, where 1 is I strongly disagree with this statement, and 9 is I strongly agree. And we define the consensus as being appropriate if it's equal or over 7 median. And that's the whole group, not divided pediatric and adult, but we also present the pediatric and adult data separately. Also, we present the level of disagreement, so how many are in the opposite side down here. So again, to go through these 7 statements here, the first couple are pretty easy to say. One is, the first one is that multi-center international efforts are essential. There's a full consensus to this, and I want to point out that there are initiatives already. Of course, we've heard from several of them here today. We'll just skip that. The second statement is that development of an international web-based high-quality clinical registry and database should be priority. Also a full consensus here again. And just as Larry was pointing out, there is a registry available already, which is open to join for everyone. And there are also other ones. Among the comments from the group or the panel here, it was also mentioned that there are other registries that are not functioning right now, but could be functional for this and leveraged to use this. Among those were the Canadian BrainWorks and the New Zealand-based EpiNet registry there. And if we're not looking at creating one registry, at least we should make sure that that it's possible to harmonize or align all the data that are in these registries. The third statement was that in addition to ongoing observational studies, an intervention trial of immunological treatment should be initiated. Again, a full consensus. I didn't mention that, but you can see the median is up here. This is the average median and for the whole group, and then the median for adult and pediatric. And this is the level of agreement here and the level of disagreement you see there. So the concerns here was that we don't really know about the pathophysiology so much, so we don't know what to study, and that it's going to be complicated to perform these studies. And this will come back in the coming questions here. The fourth question concerns sampling of CSF, whether this would be possible before, prior and following treatment. We believe this is important, and it was concerned that it might affect recruitment, but still it's probably going to be very important to look at some to be able to look at these biomarkers. So then is when it starts to become a little bit more complicated here with the statements 5, 6, and 7. So what we're saying now is that in statement 5, and these are the consensus statements, we say that it's not, as it is not ethical to randomize the placebo arm in immunological treatment trial, because we're not in equipoise. We need to have alternative study designs, and that's a fairly easy one. And then we say that a head-to-head randomized comparison between 2 selected intervention is the most appropriate form of treatment trial. And this was a statement that we based on the open question in the first round of the Delphi. Now, this is what we suggest, and you can see that the level of agreement is somewhat lower, but this is still a consensus. There are quite a lot of concerns raised over this, of course, because it has a lot of problems. If you don't have a placebo arm, if you don't have a controls, we have no way of knowing what we're really looking at. If we don't see a difference, it could mean that we have an equal effect, or it could mean that we have no effect. It could also mean that they're both harmful, but one is less harmful than the other, and we'll interpret it as superiority, but it's less inferiority, in fact. So this is it's a problem in trying to design it. The sixth, so this is the same statement, but one thing that was lifted by several respondents was also to use some sort of an adaptive randomization, posterior randomization design. And this, it doesn't solve the problem of knowing or the interpretation, but some sort of adaptive design where you could, for instance, a play winner design where you stack, you use outcome data for every patient or for every set of patients, and you reverse it and increase the odds of having the more beneficial or successful treatment. It complicates calculations on sample size, of course, but it may be more ethical. And it may, in fact, be pretty useful in this condition with a very rare disease like NORSE. And we can also use block assignment in the backwards feedback. So that would be feasible. The final statement was also based on the open question in the first round, and we here suggest that in a head-to-head randomized treatment trial, the prioritized treatments should be IL-1 inhibitors, Anakindra, and IL-6 inhibitors, so tocilizumab in this case. And you can see that there's consensus in the group. There was not consensus in the adult respondents, but there was a consensus. And of course, there's a lot of problems with this, just for the reasons we just mentioned here. Study designs and also what therapies to study. This differs a lot between adult and pediatric, partly based on the different etiologies, of course, but also due to treatment tradition. So if you look at it was almost 100% consensus in pediatrics to look at IL-1 inhibition. Also, ketogenic diet wasn't really suggested to look so much because it's considered standard care in kids. Whereas in adults, several people suggested we should look at that. Steroids, rituximab were suggested in the adult side, and tocilizumab was suggested by both. And this may be a reason to let protocols differ among age groups. So the possible study designs then that we may end up with, I've listed a few here. I hope they all have problems. So I hope the solution to how we do this is in this room here. We could do some sort of comparative effectiveness research designs. We could look at and have a standard treatment and add one treatment and see how that does. We could have a standard treatment and add two treatments, A or B, and compare them. We could do a comparative effectiveness study, A versus B, and then not comparative. And this is very close to a placebo study then. And then there are forms of using personalized expert boards reviewing each case on individualized care that could be applied to basically any one of these. The problem here is that we're not agreeing on what standard care is. And this may be a problem. We need, in order to be able to perform any of these studies, we need to agree what is standard care and what are we actually looking at. And if no immunological variables are standard care, we're going to end up with a problem, because then we're back to the placebo problem. Then they're not receiving anything, and we just said that was not ethical. One question there as well, if we should separate peds from adults, and whether it's adaptive or not doesn't really matter. One thing we discussed back in 2019, where we actually started this project, was also to use a CER design and vary, looking at one study drug and vary the timing or the dose there. Maybe a fairly weak study design, but could be feasible. It was also suggested to have a CER design with two or more different treatment plans that the treating clinicians can choose from, basically. So this will be a non-randomization that will increase the autonomy for the treating team, but of course it introduces a bias, and we won't have any controls here either. An N-of-1 trial looking at each individual patient, using them as their own controls, would be feasible. The problem, of course, is that the situation in a NORSE case is very multifactorial, and we don't really know about the natural history. So whatever we do later on in the process is going to be more likely beneficial. And also the half-life of some of the treatments that we want to study is going to make it very difficult to do an N-of-1 trial. And then finally, the possibility to have biomarker-based personalized therapies. The Texas data suggests that this would be possible, but we really, it's unsure if we have sufficient knowledge right now. One thing I just want to briefly mention is one thing that we're working on in Europe now is the idea of trying to create an individualized online clinical decision support system. So a CDS system, which is integrated with an ECRF. So what this would do is actually this would work so that we would take all the data we have. You have around 1,300 patients described in one way or another concerning NORSE. We would put all that data into a machine learning system. We could add any new studies as they come along, and then we add the state-of-the-art recommendations into this. So when you as a clinician add your patient, when you have a patient in NORSE, you add in real time, and based on the baseline data of your patient and what is currently known, you get, I don't know if we should call them recommendations, maybe suggestions. And this is what you should think about in your treatment and in your workup and so on. And as you go along with your patient, when you start workup, when you get results back, when you start treatments, and when you have the results of treatments, you put that back into the CRF, which will, or in the CDS, which will then give you new recommendations. So if you have like a 4-year-old girl or a small child, they will say, but maybe you should think about metabolic workup because you haven't done that at this point or something like that. And in the end, you also put in the outcome, of course. As this is machine learning, for every patient here, it will feed back to the initiation. So it'll learn more and more, and we can either patient by patient or in blocks validate these data. So for you as a clinician and for the patient, this is beneficial because you get, it's like an interactive online adaptive algorithm rather than having it on paper, which will be individualized for your patient. The good thing, the second good thing is that this will all feed back into the database. So this will build the biobank and be an incentive for the clinician, not only to build the biobank, because we'll have results in 2 or 3 years, you'll have results right now, but it'll also feed back. So this is what we're looking at, and I'd like to hear your opinions about this. I think we're stuck here. Can we move forward? A comment? Yeah, sure.
Eyal, if you could say something about the comment you put into the chat, that would be great.
Sure. So I'm a rheumatologist. Dr. Lay mentioned me briefly. I'm at Texas Children's and also wear a traditional rheumatology hat. And in rheumatology, for close to a decade, some of our ability or inability to get randomized controlled trials has been to have consensus treatment plans where you small work groups involved with certain rare or not so rare rheumatic diseases would come up with 2 or 3 approaches that the whole field in North America would quote unquote agree to. And then you at least reduce the variability that way. Some of this does not help with what Ronny had talked about in terms of controls, but perhaps with propensity scoring or other measures, you can treat and perhaps balance some of those confounders. But at least you know you're not getting 25 different approaches, which we used to have for certain diseases, but maybe 2 or 3, and you can think of at least providing guidance to small teams or teams with less expertise. Thank you.
Thanks, Al. Yeah, that's exactly one of the possibilities that that's up there. So we'll get to a polling just now. These are some of the questions that we had at the last meeting in Baltimore, and the first 3 there I think we passed, but the second 3 are the remaining 3 is actually what we're still left with. What kind of treatment trials is likely to be most effective for a pilot study? Are we agreeing on what drug or drugs to study, and also the outcome variables thing? Before we just to mention some possible ways forward in this is of course to start working on developing common data elements. This should be a consensus work, and this is also concerning outcome variables. This probably needs to be done in agreement with ILAE, with the status epilepticus group of the ILAE, so we develop the same common data elements as they are. To develop a clinical database and a biobank. Again, there is a biobank and a clinical database, but at least make sure that the multiple ones are alignable. What I mentioned concerning the CDS, and then the last question there is open to how, if we want to formalize a NORSE research consortium further to try to further research.
Could I make a comment?
Absolutely.
So as I remain confused. So regarding the comment on rheumatoid arthritis or the progress we've made in multiple sclerosis, we're dealing with a rather well-defined underlying pathophysiology of the disease, which one can very easily diagnose. Whereas here we're seemingly dealing, and I know very little about NORSE, quite frankly, but seems to be more a symptom than one underlying disease. We talked about having anti-NMDA receptor and different types of etiology. I think the first step, it seems to be the necessity to ask whether we're treating an underlying neuroinflammatory disease, potentially autoimmune or potentially viral, as opposed to the consequences of inflammation in the central nervous system driving intractable epilepsy. And the comments that were that it's not a disease like rheumatoid arthritis or MS, and I this what confuses me about what we're doing. And I think insight into how we can subdivide the individual etiologies of what's driving the underlying disease will be critical. Or alternatively, to say that there are inflammatory events which are common among the different etiologies, and we're trying to understand how to treat this form of epilepsy. So those are general comments that I would make, and ways of defining these diseases would seem to be the first step. I would ask thoughts on individuals who study this deeply.
Yeah. I don't know if anyone wants to comment on that. I could just say that it is a problem, of course, on two sides. The first, that we don't know the pathophysiology or the pathophysiologies behind this. It could be several diseases. The other one, that we don't have a group of patients to study. Everyone is per definition healthy until they end up in a status epilepticus. So this is, of course, complicated. I think that when we're discussing that, we're discussing NORSE here, but what we're actually looking at is probably cryptogenic NORSE cases. And although we haven't really specified that in detail, but you're absolutely right. Okay, Eyal?
Yeah, so that's a great point. Some of these consensus treatment plans are not just for arthritis, but even forms of arthritis that we don't quite understand the true cytokine or inflammatory milieu, so systemic juvenile arthritis, or even fibrotic diseases, and even lupus in pediatrics is probably not one disease immunologically, but does have a shared phenotype. So there will need to be better description of the clinical phenotype and exclusionary criteria, but pediatric rheumatology is not quite at precision medicine either, though I do agree that there's better sense of what the disease states are, but not a great sense of what the underlying cytokines are. And we're still sort of hit or miss, even in juvenile arthritis.
Right. Any more comments there? Yeah, from Lena. Oh, okay. Comment from Lena.
So I was just saying that I think intervention studies are very reasonable because it's likely that we have a final common pathway, and if we wait for etiologies, we won't get them for a while. I don't think it's right to wait.
Right. Beth?
I would agree with that.
Yeah. Hi, everyone.
Yeah.
Hi, everyone. It's Beth Wells. Can you hear me?
Yep.
Oh, good. Hi. I just put in the chat that, with our overlap in neuro-oncology and neuroinflammation, we're participating in a trial through PNOC of DIPG. There's a personalized medicine expert panel using EG, and we think that approach, could be applied here. Interesting to look at together. And so it's, it's different from a traditional trial in that, the study question is really whether the novel biomarker, in this case, cytokines, informs the expert panel's decision at key points, and then, and then whether the treating team chooses to take the advice of the expert panel. So
that too is a very interesting approach. I agree. So for those of you who haven't answered the first question, if you were, please do that. You can see. I'm sorry, please.
One comment about the ethical challenges of defining a trial would be that different latencies of introducing treatment can be utilized. So we have a unfortunate situation here where the disease is prolonged. So unlike, say, some emergency in the emergency department, you could introduce a drug at day 7, day 10, day 14, and really look at the time kinetics. I think that gets to specifically what are the outcomes in a study of interest? And because we are treating these patients and often suppressing the disease, there's a lot of influence by the treating providers in terms of, when the episode of status would end, right? You'd have to stop anesthesia to sort of determine that. And so if you had a survival analysis or time latency to recovery, you could look at that. It just requires some strict rules about trying to liberate the patient in a sort of similar way between centers.
Yeah, thanks. Okay, so we'll try to proceed. There was a majority answer that the CDS system would be feasible or is interesting. The second question, this is the second question. Concerning clinical trials, do you in this group perceive that it would be better to separate pediatric and adult in order to achieve agreement on what is standard care? Or do you think it would be better to try to pursue a joint clinical protocol? Yeah, it's really a battle here. It looks at the separationists are winning here as well as in the world in general. Okay, so there are arguments for both sides, obviously, and differing views here, but the majority is thinking that it would be easier, probably, or better to separate pediatric and adult in studies. All right, the third question, which is a very, very difficult question, and we won't have time to talk to because we're going to switch into discussing some lab issues as well. But I would like you to see what the group thinks when it comes to study design for a clinical trial. What do you think is most feasible at this stage? We were discussing a CER design with 1 or 2 interventions. Or number 2 is a 1 intervention and variation time dose. The treatment plans that Ayel was referring to, an N-1 design, or none of the above. I was thinking of putting an option with none of the above, and I have a great idea. I'm hoping that there's one. Exactly. Yeah, there's. Okay, so again, either a standard CER design with 1 to 2 interventions, or having this the treatment plans, which is appealing in many ways. And the majority is there, so maybe that's the way to pursue. Any comments on this before we switch over and discuss? We have only a couple of minutes or 10 minutes or so to discuss some lab issues as well, or some lab experimental data. So I'll leave it to Nicholas to continue.
Well, thanks everyone. This is going to be very, very quick. On this slide, I've summarized the Bergman version of what we know about.
One comment before you go on. Take more food. There's still food in the back of the room then. Help yourself.
That's fine. So on that slide, I've summarized the Bergman version of what we think we know about the pathophysiology of NORSE and FIRES. This was much better summarized yesterday by Krista and Olga at the SIG. So basically what we're seeing is happening is based on very nice studies dating back down to 2012, is that there's some minor trigger infection, some virus possibly, that in a patient that might have a genetic predisposition to inflammatory overactivation. And this is the data from the Mayo group showing polymorphisms in the IL-1RN gene. And that leads to the overproduction of CSF inflammatory cytokines, CXCL10, interleukin-6, 8, and so on, some of them being known to increase neural excitability, and that leads to status epilepticus, which is refractory, but might be responsive to these interventions like tocilizumab and the ketogenic diet, and then seizures, and kind of create a self-perpetuating loop through inflammation. So this is what we know, and everything we are trying to do clinically is based on that, using steroids, using actinides and tocilizumab. But clearly, the pathophysiology is far from being fully understood. So the first question, and actually, I think I already answered the question myself. This is a very blunt and naive question, which we have asked ourselves over the last few months, and which keep us awake at night. Is do you think we know enough of the pathophysiology in human to create an animal model that is well informed enough, so that you can ask meaningful question in this model that could then be translated into clinical valid information? So yeah, please take some time to finalize your responses. All right, I think we've reached, the poll is not going to change. I guess the results are going to be very similar, even if other people vote. So yeah, that's a problem, right? We desperately need an animal model if you want to increase the throughput in identifying drugs that might be helpful in NORSE and FIRES, yet we don't know exactly where to start. So the next question, it's actually an open question. It's not a multiple choice question. It's a word cloud. We're trying to gather idea from you of what we should learn from an animal model. So you just not pick response, just put 3 words in the application, and we'll see what comes up. Can be anything. Can be anything. Which drug works? What's happening in terms of cytokines and so on. So please enter. Single words, a couple words, not long sentences. It's not going to work. All right, keep them coming. It's great. All right, I think we almost reached a final version. We'll take a picture of that so we can keep all the IDs. So it seems that it revolves around a few things. Therapy, of course, predictors, cytokines, biomarkers, predictors have been the same thing. So therapy, which drug works best, ISO robust somewhere, mechanism, effective treatment, clinical response. So yeah, we're looking for something that will inform us on which drug works, but maybe not only on which drug works, but how it works and how we can predict that these drugs, which actually might be feasible. We need to talk with the basic lab people involved in this to see if we can come up maybe with not a model that replicates the disease, but a model in which we can try the drugs that we believe work in the disease and understand if you can predict how they work and if they will work. Cool. You've taken a picture? Yeah. All right. The two. Okay, cool. So the 2 last questions are very, very similar and close to each other. Whether it is through the biobank, the one that's currently running and the one that started at Yale, or whether it is through future trials. One of the questions is how much samples we think we can collect. And this actually leads to 2 different questions, is what we feel is important and at the same time what we think is feasible. Thanks to the generosity of the Wong family, we're going to be able to collect samples from 200 patients in total, but at the end of these 200 patients that the funding is actually over. You're not... That's right, right? Yeah. So we have to find a way to pursue the effort beyond these 200 patients. And we might need to be careful about the money we want to spend in collecting, shipping, and holding samples. So we need probably to be, at the moment, we can afford to be extremely generous in collecting as many samples as we want from as many actually organs that we think might be useful. But at some point, we need to decide and focus on which ones seems to be the most relevant. Yeah, so you're already answering the question. So seems like CSF is a no-brainer. Serum DNA, interestingly, 50% of you think that DNA is not really relevant. So okay. Brain tissue, if you can get them. Not a lot of interest in PBMCs, sponge skin biopsy for iPSCs. These are things that We may be doing in the registry and the biobank, but not sure yet. So it's interesting to hear that you guys might not be that jazzed about that. And then the next question about the same samples is, what do you think is feasible? Either through a observational funded study or even through a clinical trial where we would collect samples at the same time that we would assess the efficacy of different study drugs. So what would be feasible and what is not feasible? So what is difficult to collect among those samples? Yeah, brain tissue obviously is going to be reserved to the few cases we get to biopsy and autopsy. CSF, interestingly, a third of you say that CSF is difficult to collect. So we have to address that because at the same time, all answer that this is a very important sample. So we need to find a way to make this acquisition as streamlined as possible. Yeah, and the rest seems pretty easy. Okay, good. EEG recordings and brain MRI, pretty simple. But these are things that we haven't considered yet, or that we are considering, but haven't thought about how to do it yet. So it seems that all of you believe that it could be easily done. So that weighs in favor of us trying to do it in the future. All right, so this was my last slide. There's still a couple of slides. Yeah, so these are the algorithms that will be available in the Delphi article once it's over. All right, I think we're done. Thank you very much, Larry.
Thank you, Ronnie, Nicholas. So technically, I think we're already 2 minutes over. So obviously anyone can leave if they want, but I did want to open it up to just any further comments from anybody about anything.
I think there's just so much bias in these prospective registries, especially when they're small, they dominate. And even if we can start doing randomized studies of drugs in small populations where we get to look at the change in certain biomarkers, inflammatory biomarkers, I think it'll give us a much more sort of unbiased view as to what drugs do rather than the patients for whom we choose to give drugs to. I think those are two different things. So we may be a little far away from clinical trials to assess efficacy, but biomarker studies, I think there's certainly an urgency there because collecting more data that is informed by, these bias selection of treatment, I think is a little bit risky actually. That may lead us down a path that is unfortunate.
So this idea is only partially baked, but I understand the desire to do a clinical trial. At this point, I don't think we know enough about the disease to formulate that trial. But one thing we could look at that would be relatively simple to do would be what's the value of completely suppressing seizures versus allowing a small number of seizures. We could have an adaptive trial design that would, I think, fairly quickly establish whether there's an outcome difference in so doing, seeing that some of the drugs we use like propofol have so many effects that are immunosuppressive, other potentially negative effects. I'm not sure how to design this yet, but it is something we could probably agree on and accomplish while we're trying to understand immunology better.
So also a comment regarding the trial design. I think here we have to think out of the box. There are innovative designs such as counterfactorial outcomes, G estimates, which allow control for treatment adjustments throughout the time. So we have a lot of time-dependent covariates, which we have to take into consideration and cannot simply look at the single outcome endpoint and do the Kaplan-Meier. So I think that needs a lot of thought, and I agree with Tom. This is the unifying hypothesis, what we have, the immune, triggering an immune response which creates all the trouble coming later. But what we do over the time by treating the patients, there are so many things going on which we have to control for. Therefore, we cannot use a standardized classical trial design. We have to go another route, as I mentioned.
If anyone on Zoom has a question, you can just unmute yourselves. Anna Maria?
Yes, just a comment about the development of animal models. I fully agree that since we do not know the etiology, and until we will know the etiology, then the animal model cannot reproduce the etiology of the condition. What the animal model might reproduce, if it is cleverly designed, is a condition where you can couple a neuroinflammatory response to a kind of latent hyperexcitability that could mimic gene variants in coding for proteins that are chiefly involved in hyperexcitability phenomena. And by combining these two phenomena, there will be a model that we can validate for its response to or refractoriness to anti-seizure drugs for which there is refractoriness in the clinical condition. So the model, in my view, could give insights into novel treatments for the clinical translation, and in addition to provide potential predictive biomarkers of a therapeutic response. And incidentally, Anakindra, that appears to be effective at least in part of the patients, has been developed in the preclinical model of acute seizures to start with. So I think that the model is useful, but we have to ask proper questions to the model, and until we know the etiology, we cannot reproduce it faithfully, except maybe for the autoimmune etiologies, where then one can focus on a specific cause of the syndrome.
Dr. Heffler?
Yeah, my comment would be that of all the things that are being done, I think one of the things we're learning in immunology, I'm just writing a program project grant on this, is that mutations in checkpoint inhibitors, not homozygous mutations, which lead to childhood diseases, but heterozygote mutations. For example, in TIM3, one of the major checkpoints, leads to a hyper macrophage syndrome. There's a microglia, the macrophages become activated, and the frequency is about 1 in 100. And other heterozygote mutations in checkpoint inhibitors may be occurring also. So I really hope that the whole exon sequencing with a particular focus on immune pathways related to immune regulation, in particular checkpoint inhibitors, may give insight to what may simply be a hyper microglia syndrome in relationship to, as suggested earlier, with common viral infections, something which we're just beginning to explore in the field of immunology.
Great. Sounds like relatively low-hanging fruit to explore.
Exactly.
Dr. Nashif?
Thank you, Larry. Just in relation to the comment made by the audience, by somebody in the audience, saying that we don't understand enough to be planning treatment, I wasn't particularly thinking about new treatments that are not on the table. I suggest that the trials are done to test the treatments that are already being given in many centers. So they're being given, but the data is not out there. So we should really at least design studies to test them.
That's a good point. We may not know enough on what to study, but it's already being used, so we should study it while we're using it at least. Are there more? Eyal, did you have another comment?
Well, I think since the epilepsy community often has partners, I do think policy statements that in the US will allow centers to get whole exome sequencing more easily on the clinical side may allow some of the people in the room to sort of be able to get the data for the researchers that is clinically relevant and potentially actionable if an institution can do clinical whole exome sequencing quickly enough. And there are some centers out there that are doing this for many other catastrophic diseases.
All right, fantastic. Well, thank you all for staying late. Thanks to all the Zoom participants, and thanks everyone here for coming in person.